Okay, I think we might be live. Hello, everyone, and good afternoon. Welcome to the session on data sciences and brain health across the life course. My name is Kristen Flagel. I'm the lead scientist for the Sinapse Research Network. And this is a collaborative session today with presenters representing 3 different Scottish research pools, and as well the Scottish Dementia Research Consortium. And we've recently been discussing among these different disciplinary representations where there might be opportunities that are really exciting to move things forward by integrating data sciences into the brain health research areas of neuroimaging and neurobiology to address some really significant challenges around early detection of neurodegenerative disease. And I have just a few slides to introduce the context for today's session. What I am showing you here is an image that's taken from a new website that the research pools have collaboratively built and launched. The URL is there in the bottom corner if you're interested in exploring yourself. But what you can see here is a sort of map of Scotland's research and innovation ecosystem. You all will, of course, be familiar with Sicsa as a research pool. You're probably aware that there are other research pools around. As I mentioned, I'm the lead scientist for Sinapse, which is the medical imaging research pool for Scotland. There are other aligned initiatives and organizations around the country whose work comes into contact with the research that the individual pools are doing, and what the pools have been moving toward is more integration, more interdisciplinary activity, and a lot of this is captured in this new website, so I welcome you all to check it out. So today's session is led by Sinapse, the medical imaging research pool. We also have some work that's going to be presented on behalf of Sulsa, which is a life sciences research pool, and Sicsa, of course, the computing science and informatics research pool. And we also have a presentation from the Scottish Dementia Research Consortium. And you're going to hear about work that is driving toward bringing together experts from these different disciplines. And we see opportunities for other parts of the Scottish research and innovation ecosystem to join in as well. So we're really only in the early stages of these discussions, and that's one of the reasons that we're excited about this session and the opportunity to have some dialogue with attendees today. We've had some early discussions with a few of Scotland's innovation centres where we see natural opportunities to link together. So today's session begins with 4 brief presentations, all addressing multidisciplinary challenges in the early detection of neurodegeneration from 4 different perspectives. So on behalf of Sinapse, first we'll hear from Dr. Jerry Thompson from the University of Edinburgh. He's going to be presenting on brain imaging data. Then we'll hear from Professor Bettina Platt from the University of Aberdeen. She's going to be presenting on translational multidisciplinary approaches to the same sort of questions that Jerry investigates by doing brain scanning with humans. Professor Mike Chandler from Heriot-Watt is then going to present on some data science approaches that he uses to model topics and visualize data in really large-scale data sets. And finally, we have a presentation from Dr. Graciela Muniz-Terrera, and she, from the University of Edinburgh, is going to wrap up by discussing some of the challenges that remain for delivering clinical impact from predictive models of brain health and dementia risk that can be built using some of the data that we have heard Jerry and Bettina talk about. So we're really excited. After these 4 talks, there will be time for discussion, and we would encourage a lot of interaction. So please do type your questions into the chat window that's available. Here are some thoughts that you might have in the back of your mind as you hear today's presentations. And if this is an area that is of interest to you, we would like to welcome you to sign up for a new mailing list, which is launching today. There's an email there's a URL there that you can follow to register yourself to join this mailing list. All of today's presenters are on the mailing list. And as I say, we're working to build a community together that hopefully will be taking some action moving forward from the early stages of work that's going to be discussed today. So that's it for my introduction. And I'd like to hand over to Dr. Jerry Thompson for the first talk in the session.
Okay, thank you, Kristen. I will just share my screen and hopefully we can get going. So shout out if it's not coming over or if I don't sound clear enough. And okay, so my name's Jerry Thompson. You can see there I have sort of two hats, as it were. One of them is an NHS hat, which is a diagnostic neuroradiologist at the DCN Hospital in Edinburgh. And the other hat is an academic hat, which could be roughly summarised as being an imaging scientist. And this is quite a challenging talk to give in some respects, because it's not too much time to deal with three quite big components. One is brain imaging data, one is dementia, and one is biomarkers. And I've tried to not assume too much sort of prior knowledge amongst the community that we have here. Now, the talk is in itself it would be far too long, I think, for the time slot. But what I decided to do was, rather than trim it really down to bare bones, I've left some slides in there that I may kind of go past or gloss over, but then it's fuel there to have the discussion. If anyone wants to ask a question or highlight or flag something up, then there's something I can refer back to. So what I'm going to talk about, I'll do a bit of background about Alzheimer's disease and dementia, just to set the scene, something about biomarkers and this sort of omics term that is something I get asked about quite a lot. What I do currently for clinical imaging practice with my NHS hat on, which is sort of what you might term qualitative imaging. So it's my interpretation of the pictures that we see. The role of my other hat, which is the imaging scientist, which is more around the quantitative side and what we term radiomics, which is one of these omics that we often talk about, and how that relates to biomarker discovery from what is crudely termed advanced imaging, but that kind of means stuff that we don't do on a day-to-day basis in the NHS. And then a bit of a summary, and perhaps the key thing of this whole talk is that we do need this wider engagement, because the biomarker discovery is really going to be predicated on a huge volume of quite complex data. You know, as imaging scientists, as we generate more data, it requires more cross-domain expertise and complexity to deal with that and make something meaningful from it. So just to clarify, Alzheimer's disease is a type of dementia. It's the most common type, but the majority of cases are Alzheimer's. But dementia is just a general term for symptoms like your memory's going off or your reasoning skills or other thinking skills. And it's just quite clear to, people often use them interchangeably. It's not quite the case, but by and large, AD is the biggest challenge when it comes to dementia. The cost currently is estimated worldwide at $1 trillion a year. That's direct and indirect costs in terms of the management and the impacts. We've had limited progress for both symptomatic treatments and disease-modifying treatments in the last couple of decades. And it may be the case that current detection that I do on the NHS side may actually be too late to do anything meaningful in terms of changing the disease course, which is why we need to broaden the suite of biomarkers. So there's growing evidence that this disease that I usually see quite late starts earlier in life, perhaps middle age, and there are some theories that it begins even earlier than that. Just some words just to say this. Preclinical can have two meanings in medicine. Some people use it to refer to animal models of disease. In the dementia and AD context, what we're talking about is a disease process that's underway, so there might be microscopic changes, but it's not apparent to the person or to your friends and family that there's something underlying that's going on. Prodromal is beyond that. That's where something's not quite right, but it's not enough to classify or fully qualify as having the disease as we call it at the moment. And part of that prodromal phase is what we call mild cognitive impairment. So that's people that turn up to the GP saying, I keep misplacing my keys, or I forgot where my car was parked in the supermarket. I mean, that's a crude sort of reduction. And AD is Alzheimer's disease. And the challenge falls into sort of three main domains here. Identifying the early determinants, what goes into your cognitive health, your brain health. Identifying sort of early treatments that will delay the onset of your decline over time, and you can see an age scale down here, starting in middle age. And identifying treatments to relieve symptoms and slow progression. So if this is the time course of someone with dementia, and they see there's a steep decline into the 7th, 8th and 9th decades, what we're trying to do is intervene to get more of this kind of green line. But what is important, and if we think about context of research, is that this is a long-term research requiring process here. You're not going to get 3- and 5-year grants that are going to solve all of this at once. We'll have to have cohort studies and follow-up. But engaging with sort of data scientists and related disciplines can really help to extract as much information as we can out of the data sets that we have at the moment and can get. And a lot of my NHS work is predicated on this kind of what happens to the shape and the structure of the brain. And you can see that the brain looks kind of normal here. It starts to waste away in certain sort of characteristic patterns. And it actually looks a bit like a walnut. And what's going on under the microscope is a long-established thing called the BRAC stages, which really relate to the accumulation of two main things. Tangles, which are made of tau, a type of protein called tau, and amyloid, which is a different protein, and they have a spatial and temporal progress through the brain as it starts to fail, and that relates to how people with Alzheimer's present. And these two things are important from an imaging point of view, as I'll show you. And the way that we currently classify it uses this ATN framework, and what I'll just point out is that there are various things that can go together to give you different type, classifications that are, they're quite reductionist from a clinical and a scientific point of view. There are only a few different groups, but you can see that we use it to mainly determine whether you've got Alzheimer's disease or whether you've got one of the other things that might link in with it. So again, it's fairly crude. It's not very specific, and it often happens at a time when you've got a lot going on already. Biomarkers, that's very simply a portmanteau word for biological markers. And the dictionary definition of this, which isn't perfect, is naturally occurring molecule, gene or characteristics for which a particular pathological or physiological process disease can be identified. Effectively, what it means is there something we can see or measure that has a meaningful relationship to what's going on with a disease process? That can be an imaging test, a cognitive test, a questionnaire, a behavioural study, a blood test, you name it. If you can extract information that may relate to a disease process, then that can be a biomarker. And how useful it is, it comes down to a balance of the cost, the accessibility, the tolerability. Is it a sample of blood versus a non-invasive thing and filling in a questionnaire, and how well it performs. Basically, how good is this compared to just tossing a coin? What additional information are we getting? And there are different types of biomarkers, and I tend to think of these more with these italic phrases than the kind of sort of Greek-derived and Latin-derived words for them. So diagnostic is what's wrong, what disease have you got? Prognostic is, well, how long have I got before X happens? That may be death, it may be decline to the point where I can't work. It's those kind of things that we look at clinically. Predictive, how will my disease behave? There could be two people the same age who both have a diagnosis of Alzheimer's disease, but some might take longer to reach certain time points than others. This is a really interesting one that imaging we use increasingly for is pharmacodynamics, which just means what drug do I choose and what dose should I choose to balance risks and benefits and side effects. And the response, which kind of goes in with the pharmacodynamics, which is, will my disease respond to treatment? So that's kind of, you can work out whether a drug will get into the brain, which is a good thing to know if it's going to work. And then it's, well, Mrs. Bloggs, will that work for Mrs. Bloggs and not Mr. Smith? And that's what this kind of response biomarker helps us with. And if I put that in the context of imaging, this bit is what I do in the clinic with my NHS hat on. So anatomical magnetic resonance imaging and a bit of FDG, sort of glucose metabolic PET. And imaging science at the moment, the commonest things are amyloid PET and tau PET, which are basically non-invasive imaging techniques that look at those BRAC stages that I showed you earlier. And when we talk about biomarkers over time in dementia, we have this concept of where can you actually detect that it's abnormal, and then what happens over time till you get frank, severe dementia. And you can see that we have a suite of different biomarkers in the fluid that surrounds your brain and spinal cord, which requires a spinal sample, what we call a lumbar puncture that's quite invasive. Amyloid PET, which is an injection of some radioactive material and sitting on a scanner for an hour. This combination that I've said before about MRI and FDG PET, which is much more widely available and a cheaper than amyloid PET. And cognitive impairment can be something as simple as filling in a questionnaire or a psychological survey. And they have different sensitivities to what the disease process might be doing. Now, omics, this is the easiest way I can break down omics is that, and to use, if you like, a sort of computer science analogy, the genome is the instruction, so that's the program, and the phenotype, or sorry, the phenome is what you get. So the genotype is your individual genome. So I have a genome, Mike's a genome, Graciela's a genome, but our genotypes are different because that's our individual genomes are different. And the phenotype is what you get out of that. So I am male and I have red hair, and Graciela is female and she has brown hair. And the phenotype is sort of conditional and dynamic, which responds to aging, and during the course of a year, even diurnally, your phenotype can differ. And so phenome means all of the things that happen as a result of your genome and what's called the epigenome, which are things that you can turn genes on and off with. Now, part of the phenome is, so transcriptome means how do we get the message out of the middle of the cell into the rest of the cell we can do something with. Proteome, that's the proteins we make from the transcriptome. The metabolome is all the metabolism in the cells. Synaptomes, the linkages in the brain. Inflammasome is the inflammatory cells. Basically, biological scientists like to stick any word they can at the front of ome, and that describes what, some concept of how your instructions in combination with the environment lead to you and what's going on with you. And a really key thing of this is, so from my point of view, when I image, I'm looking at what's called the radiome, and this is just one part of a huge cloud of stuff that all links together to describe what an individual experiences and what they will demonstrate when you look at them. And a really key thing for this is how it changes over time because it's dynamic. So the role of the radiologist is pretty straightforward. If you go to your doctor tomorrow saying that you're forgetting where things are and misplacing your keys, you'll probably get a CT scan. That is more to say you don't have anything else going on, like a brain tumour that may be pressing on a bit of the brain that causes similar symptoms. Then you'll get an MRI to make sure there's definitely not another cause, because it's a bit more sensitive and specific than CT. And what we tend to say is there a pattern of, the brain shrinkage or strokes or something that we can tell you what kind of dementia you might have, because there's Alzheimer's disease and there's others. And then we have PET and SPECT to fall back on. And I use that if your CT and MRI look normal, but your clinician is telling me, no, there's definitely something wrong with this person's memory. And I look at things that precede your brain shrinking, which are things like what's the metabolism like, what's the blood flow like. And so this is, in a crude way, is I look to see if this structure here, which is your hippocampus, that's crucially involved in memory and memory storage and processing. So you can see that over time, people can lose volume in the hippocampus, and that we give it a severity score. The other bit that's involved is this bit at the back of the brain in the parietal lobe, it's called the precuneus, and that shrinks over time. So the severity of these two link together to give me an idea of how severe your Alzheimer's disease might be. Okay. And then we have another component here which looks at how much if you've got high blood pressure and diabetes, you can get little insults in your brain where it won't function, the wiring won't function quite as well, and that can be a compounding thing. And I take all of those together and I try and work out, these are biomarkers for me, structural MR biomarkers, and I try and work out what kind of dementia you might have. And that's effectively my job. If I use the nuclear medicine we talked about, so the FDG, which is the metabolism, you can see the same bits of the brain that we get the shrinkage show up as what we call hypometabolic. in Alzheimer's disease. If you have a different kind of disease called FTT, it's at the front, and that's another tool that I can use, a biomarker that I can use to describe that. And here's what amyloid looks like. So pathologists under the microscope will see amyloid build up in the grey matter in the brain. That's a healthy person where you see very little. And in different diseases, you can see you get a build-up of this stuff. So it looks what we call hot. There's high uptake on a PET scan. This is a bit of a problem though, because about 1/8 of people in their 60s up to half of people in their 80s that are cognitively normal will still show this as being positive. And that's where the performance characteristics of your biomarker come into play. If it's not absolutely sensitive and specific, which almost nothing is, you need to build it in with other biomarkers to work out what it all means. And that's where the data science can be key. And here's just an example showing tau and amyloid together, you know, as a pair of things to try and work out what's going on without having to take a sample of anyone's brain. So from an imaging science point of view, my question is, can I put some numbers on that? And the answer is yes. So each of those images that I've shown you an example of is basically some sort of matrix, or occasionally we have a frequency domain spectrum. So that's just a numerical thing, and they're all spatially co-localized, so they can be co-analyzed. And depending on the resolution, each of those data sets can have millions of data points for a single individual and visit. So then if that person shows up 3 or 4 times over 2 years, that scales linearly with the amount of data that we've got. And then we've got additional techniques and complexity that each have their own modeling requirements. So each of the PET tracers is slightly different, the diffusion imaging, all of those other things is different. So we deal with quite a lot of data ourselves as imaging scientists. But then if you compare that across a huge group of thousands of individuals who all have other things like electrical measurements in the brain, activity monitoring, speech fluency, clinical tests, that's just a huge amount of data to deal with. That's when we need to bring in others to help us. And here's just an example of ways that we use a fairly straightforward algorithm to get different areas of the brain so that we can work out regional variations. And this is saying, you know, this is the grey matter thinking bit of the brain, this is the white matter wiring bit of the brain, and this is the fluid that goes around and bathes it. And just to reassure from a translational point of view, we can do exactly the same thing in like large animals. So here's a sheep model that I work with, that we, you know, it's the same. We can use these biomarkers and validate them across species. Can we improve on it? Absolutely. I've only got it, the imaging bit I've shown you today is the kind of almost the sort of bare minimum barrier to entry for what we want to do. All of this MRI and all of this PET we can acquire to answer different hypothesis-driven questions. So you can now see that I can end up with billions of data points in a single experiment that I need help with, you know, storing, analysing, doing things with that data. And then if you think there's a lot of MRI and PET, what we have in Edinburgh, this is an MRI and PET scanner that's combined. So this simultaneously can acquire a combination of these and these. So you can see the data complexity is really ramping up in recent years, and we need to sort of catch up with that in terms of how we design and deal with this. And what we're effectively trying to do here is go from just this bit, which is, can I give you as early a diagnosis as possible, to say, well, how do we give you a treatment-specific prognosis that's individual to you and follow you post-treatment and know whether it's working? But also go back to the start of the process and say, can we screen people for risk? We want to be sensitive and catch them. That don't necessarily have to be specific, but then we do need some specific tests that move on from that, and we need to do discovery science at the same time. And this is what we call the personalised treatment cycle. You measure something, you analyse it, you say, what's the best treatment for you? You do some confirmatory tests, you apply the treatment, you see if it's working, and then you test again. And there's a virtuous cycle where you just check, is that individual needing to stay on this drug? Or even, can we do analysis and be reassured and say, look, we've got a few years to wait before we have to think about drug candidates. So let's just hold fire, and this person can get on with their life and the knowledge that nothing bad is going to happen in the next decade or so. That's equally as important as being able to jump in early and do something about it if we think there is something wrong. So in summary, current radiology practice is pretty subjective and largely limited to what I think the type of dementia you might have. And even then, it may be too late to do a great deal about it. So we need a suite of these biomarkers that are, that they're readable, they're reliable, they've got acceptable performance characteristics. That's for everything from screening to early diagnosis to treatment suitability and response. And imaging science is producing, almost exponentially increasing volumes of this high quality, high volume, high quality, high velocity data. But we need this integrative data science and modeling approach to combine this and make sure that we're asking and answering appropriate questions across the spectrum of disease. And really, that's what collaborations through networks, Synapse and Sicsa will be crucial to that kind of thing, and bring the, the computation and the biology and the clinical practice together to do something useful with all of this data. So hopefully that was understandable, and we might have questions now. I don't know if Kristen wants to have questions at the end, but hopefully that was a cook's tour of everything that we were thinking about our imaging biomarkers.
Thank you, Jerry. We actually will just go into the next talk at the moment. And I want to apologize to the audience. I didn't realize I wasn't sharing my screen earlier, so I'm sorry you couldn't see my slides. I will share them again once all 4 of our presenters have finished and you can see the links that I was referring to. Next, we have a talk from Professor Bettina Platt, and I'll hand over to her.
Right, okay, so I hope this is all working and you can see my slides. And thank you very much to Kristen and the organizers for the opportunity to briefly tell you about our translational research approaches. And also thank you to Jerry for quite a detailed, yes.
Sorry to interrupt, it's Andy, the technician in the room. It's just to let you know that you're displaying your next slide as well. If you don't mind going up to the display settings.
Yes.
In the top corner.
And then go to...
Yep, display settings. And then if you just go to swap presenter view and slideshow.
How's that?
That's you, perfect. Thank you very much.
Good. Okay, sorry for the delay. Some more presenting issues. So I will be talking now, following on from Jerry's introduction, about the more experimental translational neuroscience that we are conducting, and I'll give you a few examples to actually clarify what it is we're talking about. There's always a lot of omics, as we already heard, to study different symptoms and phenotypes and biomarkers. And we hear a lot about translational, multidisciplinary, cross-disciplinary, and interdisciplinary research. And I'm trying to give you some examples of what that means for my research area specifically. So we already had an introduction to dementia, so I can keep this very short. Just to say that the key issues for dementia and Alzheimer's in particular are not resolved yet at all. We don't understand the causes. We do not have reliable early diagnosis, and there's no effective treatments, but we've heard that from Jerry already, so I can keep this very brief. From an experimental neuroscience perspective, what we're doing in Aberdeen and in my research team is we work with various experimental models of Alzheimer's disease, Parkinson's disease, and so on, so the whole spectrum of the neurodegenerative disorders, and we characterize them by means of multiple experimental endpoints. For example, behavioral analysis, how is the cognition of these mice? But we obviously also look at imaging markers and also EEG, so the recording of the brain's electrical activity. And we characterize these so-called animal models in depth in vivo, in the living animal. And then we move on and study molecular and tissue markers and try to correlate the severity of the disease phenotype, the symptoms, with the severity of markers that we find in the tissues. So the whole idea about the translational approach is that we have research models in the lab, which we characterize, look at phenotypes and biomarkers, and then compare and contrast and validate looking at human dementia. And we already heard about a few examples. So we are looking at potentially harmful genes and proteins. We are looking at environmental factors such as toxins in the environment, or lifestyle factors such as diet, which are all very important risk factors for dementia. We are then using multiple experimental endpoints, as I said before, like behavioral studying. We measure brain activity. We can do brain imaging in our experimental models. We can also look at metabolics, and we can do very detailed tissue analysis. This is then compared with what we see in the clinic and in human dementia, obviously not in all endpoints, but as far as possible. We compare our results with the cognitive test results, with brain imaging and brain EEG recordings in humans. And then we look at post-mortem tissue markers to see to what extent what we see in an experimental model actually aligns with what we see in the clinic. And overall, we're trying to close the loop to connect these different experimental and clinical domains to validate our disease models, to validate our disease biomarkers, and then, of course, to engage in drug discovery to develop better, more effective treatments. In terms of computational science, we develop data platforms. We contribute data to various online platforms where we can share with colleagues. We also collaborate with computational neuroscientists and mathematicians to develop new data analysis tools. And we try to develop more reliable and robust standard operating procedures. And this is what I want to talk about very briefly as an example. So we are members of the EQUIPPED consortium, which is led by Malcolm McLeod at Edinburgh University and Thomas Steckler at Janssen & Janssen. The idea here is to improve the reproducibility and the reliability of experimental methods. It has been found over the last decades, really, that a lot of the experimental work that we're doing in the lab is not reproducible, sometimes not even within a lab, but certainly not between labs. And the translatability into the clinic has often been very poor. So, for example, a compound that we may find is highly effective in an experimental model is often found not effective in the clinic. And we think this is to do with sometimes unreliable research practices, and that we need to be much more robust and develop standard operating procedures that we know will deliver reliable outcomes. So the EQUIPPED consortium was founded by 29 institutions across 8 countries, mostly Europe, but also the United States. And there's also a large group of stakeholders, funders, publishers, patient representatives, and so on. The idea here is to develop a framework for a preclinical research quality system that we can apply both in the public and the private sector. And the quality control system that we are developing as an online platform is, the idea is to boost the reliability and innovation by ensuring that we have more robust procedures in preclinical research settings. The second example is kind of following on from what Jerry was talking about. The holy grail in dementia research is that we have early diagnosis, so we can have early intervention. And I'm not going to talk about the different phases again, that was all beautifully covered. Just to show you an example how we brought together clinical, experimental, and computational sciences in this area. I do use imaging like MRI and PET as well, but my main approach, and more used in my lab, is to measure the brain's electrical activity. And here, because you don't measure structural things or you don't measure accumulation of any toxic protein, you measure the brain's activity, how it communicates in real time. It's a non-invasive procedure. It's very inexpensive compared to a brain scan. It's versatile because you can combine it with other kinds of, for example, cognitive testing. It's very fast and simple to acquire reliable EEG, and it's translational, meaning we can apply it to the patient and we can apply it to our experimental animal models. Most importantly, though, what we have to expect is that in dementia, the earliest signs of something being wrong with your brain is that your neuronal communication and the connectivity between brain areas will slow down. And therefore, you need methods with high temporal resolution that can actually in real time follow how you are thinking or solving a cognitive task. So that's why my money for early diagnosis is on the EEG. We have conducted a huge number of EEG and sleep studies in dementia research in our animal models, and I don't really want to go through all of them, so don't worry about that. What I want to talk about is how we've teamed up with computational scientists and mathematicians to develop new ways of looking at the EEG and to improve what we can actually do monitoring the brain's activity in a behaving mouse or in a patient that is solving a cognitive task. I won't go into the math either. I just want to very briefly show you an example, what exactly I mean when I'm talking about that. So when we record the brain's electrical activity, the EEG, what we do is we construct a so-called EEG power spectrum, which shows you the intensity of different frequencies that basically make up the electrical activity or the EEG trace that you record from the human scalp. So here in this graph, you can see an EEG power spectrum from healthy controls and from a small patient cohort that was diagnosed with mild cognitive impairment, suspected Alzheimer's disease. And without going into too much detail, we can see the frequency composition of this EEG spectrum. And what you can see is that in the red line for the Alzheimer's disease patients has a higher content of slow waves here in the lower frequency range, and it has a smaller content of frequencies in the so-called alpha beta range between 50 to 20 hertz. So this was an EEG study that we conducted using novel computational tools developed by our collaborators, which gave us a very robust readout with a very low variability in this cohort. We then looked at many different animal models to see which one is actually a good match, which one translates, which one has face validity, as we call it. And from looking at a multitude of animal models, we found one which nicely recapitulates the rise in the lower frequencies. So the PLB2 APP mouse is the Alzheimer model, the PLB1 WT wild type mouse is the control model in black. And you can see that very similar to the human condition, the APP model has enhanced lower frequencies and reduced higher frequencies. So all in all, without going into too much detail, by trying to combine the clinical data and the experimental data, we could identify the animal model which, in terms of this specific biomarker, has a nice matching phenotype in that the animal model shows a comparable Alzheimer-like EEG slowing and therefore has a reliable face validity. So in conclusion, it is very obvious that dementia remains the leading unmet health concern for our generation, for our societies, for our aging societies particular. We are still far away from having methods for early diagnosis. We still really don't understand fully the causes, which also was quite clear from what Jerry was saying, in that we see a lot of the pathologies in healthy elderly people, and effective treatments are also not available. So therefore, in the context of this conference here, I would suggest that we need more robust and validated research methods. We need to validate our experimental approaches and compare with what we see in the clinic, with what we see in the patient. And we need to have a much more robust attempt to network, to exchange data, and to align our research domains in order to solve this problem. So that was me. Just wanted to thank my group and you for your attention. Thanks very much.
Thank you very much, Bettina. So we are back to keeping to time. So I would suggest that we move on to Professor Mike Chandler next. So speaking of what is needed in terms of the data science side, that is a nice segue into the presentation he'll be giving us.
Okay, thanks, Kristen. Can you see my screen okay?
I can.
Great, okay. So thanks very much to Jerry and Bettina. Rings lots of bells. Actually, I started off life doing a lot of image processing and power spectral density estimation. My power spectral density estimation was actually 2D rather than 1D. It's a very tricky field, so very nice results there. After about 20 years of doing that, yes, I am quite old, I started to get drawn into some strategic committees in terms of the research councils, and I discovered that the research councils and these strategic type committees didn't really have very many tools for looking at the portfolios they've got, and I got involved in quite a lot of networks, and I discovered that the networks as well didn't have very many tools. So for about the last 10, 15 years, I've been trying to produce tools which will help networks and strategy committees and tools especially which help communities form. So I'm not going to talk about image processing, and I'm not going to talk about power spectral density estimation. I'm going to talk about something completely different, topic modeling, which is one of the tools which we've been using quite a bit recently. First part of my background is I'm a theme lead joint with Yasha on the Sicsa data science. And the technique I'm going to talk about, Yasha has a lot of experience about, so I'm hoping he's not going to ask me any awkward questions at the end of this. Right, so you can think about, in terms of data science, you can think about it in terms of classification, prediction, and clustering in general techniques. And I'm going to be talking about clustering, and it does have, I think, a link to sustainability, the types of things which I'm talking about. So nowadays we have many large knowledge bases comprising of thousands, hundreds of thousands of documents. And the question is, how do we sustain and reuse this knowledge? And how, in particular, do we structure these large knowledge bases? Now, you may think it's quite simple because here I'm showing some grants from MRC, etc., and they already have their taxonomy, so what's the problem? Well, the problem is that you don't have an accepted taxonomy across, for instance, the research councils. So when they started their COVID-19 grant initiative at the beginning of this year, this initiative is right across all the research councils. And after about 3 months, people started saying, so what is it you're funding with COVID-19 grants? And the answer was, we can't really tell you because we don't have a taxonomy which is accepted by all of the research councils, the 8 research councils which are involved. You couldn't exactly use MRC, a taxonomy, to describe EPSRC, for instance, because EPSRC would really object and vice versa, etc. So it's quite a political problem. So how do you do this? Well, to the rescue, rushes something called topic mapping. So we can take, say, 10,000 grants, which is the current grant set across all of the research councils. We can pour them into a Java pipeline, and out pops this diagram here. This is what we call a topic map. And we use a technique, topic modeling, due to a very smart guy called Bly, 2003. And we generate these topics automatically. So it doesn't matter where the grants actually come from. The grants can come from anywhere. They can come from they can come from BBSRC, EPSRC, etc. For one example. Pour them into the pipeline, and what you get is you get this, all right? And when you have a look at this, these are also, the visualization side is very important, because if you don't group them together, it's difficult to make sense of this. So down here, for instance, what we've got is some science type stuff. Over here, we've got some data analytics, etc. And it kind of neatly breaks down into areas. All of the map, the topics, the mappings are completely automatically generated. So if we have a look at this area, which might be of more interest to the audience here, and see what is a topic. All right, so what we'll do is we've got a topic down here which mentions brain. So if we have a look at this, then we discover that the topic is in fact not just the 3 words which I've shown you here, but is a distribution of words across across the corpus of words. And hopefully these words, now I haven't exposed this to anybody who actually knows about this area, so I'm just hoping somebody isn't going to shout out and say, this is rubbish. Yes. So hopefully these words will make sense to you. I'm just curious about this mouse up here. Presumably that's not a computer mouse. This is a real mouse which is used in experiments, etc. And this topic is completely generated from, so here we have a list of some of the relevant grants, and if we look at one of these grants, then basically what we do is we use all of the words in here. We just, all we need is the words. We don't need any structure within inside there. We just take all the words within the 10,000 grant summaries, and what that does is it produces these topics. And then what we've done, once we produce the topics, is you can produce the mapping back onto the documents, or the grants in this case, so you can look up the grants. So it does 2 things. It simultaneously generates these topics, and it generates the mapping of topics to documents, and the only input that you need is the documents. You can of course do this in multiple levels, because whenever we generate these, they instantly say, Mike, it's not quite the right number of topics. It's either too detailed or it's not detailed enough. And therefore, what we can do is we can generate at several levels and then look at one super topic and see what we've got in the way of subtopics. We can also look at things like trends and what's happening in the area and that sort of thing. And here's a topic from it's a subtopic. And so hopefully this subtopic is much more detailed than the previous topic that you actually saw. And here are the grants which this subtopic is linked to, etc. Okay. So the main thing is that this is a technique where the topics, the document mappings, and the layouts, they're completely automatically generated. You give us a document set, which is just 10,000 documents, 1,000 documents, 100,000 documents. We stick it into the pipeline, and what it does is it does everything and generates these multi-level topic maps. So it's completely automatic. One of the reasons why that's very valuable is because it's very fast. And so we did a topic map, for instance, of COVID-19 documents. It's also politically agnostic. There's no one organizational person which has used it. Okay, so using the data, how is it used? The most common way that it's used is these topic maps are actually used by people to help them, and we've actually done several experiments with groups of grants administrators, etc. And what they the overriding outcome is that they say yes. It gives them an objective overview, and it makes them realize that their individual views are very, very biased, and they find these very, very valuable for cooperating within networks and doing things like horizon scanning, community collaboration, things like that. And that's why I hope that it will be of interest to your network. The documents themselves, so here I have the documents going down here, and each one of these documents has a fingerprint, all right? So these are the topics which this particular document here, which is about early warning sensing systems for infectious diseases and antimicrobial resistance. So each one of these documents has a fingerprint, all right? So now it becomes maybe slightly more interesting to you, because what we can do is we can turn unstructured text into a feature vector, which we can then feed into your machine learning. So instead of feeding in an image, which is a very structured set of data, i.e. all the pixels, what we can do is we can replace those numbers with these numbers here, and we can use all the same classification tricks that you use for image processing to start classifying individual documents. And so what I'm interested in, what we haven't done yet, is to use these sort of techniques in fingerprinting in order to augment the information that you might get through imaging, etc. Okay. You can also see how these documents evolve. So here's a document, a financial report, and you can see that it starts off with the financial statement. It goes on to talk about assets. It talks about income for the year, and it also talks quite a bit about risk here as well. And this is the document evolving as you're going down here. So you can get a view of what's in the document, how it evolves, what it's talking about. So just some quick examples. We've got famous Brexit mat, which I thought everybody would use. However, it's a political hot potato and nobody will use it, despite me contacting various high people and the UK academic Brexit committee. They didn't want to know anything about it either. So what we have here is 28,000 UK funded projects, 8,000 EU funded projects with UK collaborators. We did a topic model of absolutely everything. And what this does is it allows you to see what will happen if we lose the EU funding, all right? And we will lose something like access to 60% of our research on viruses, for instance, yeah? So you can understand why this was a bit of a hot potato. Anyway, another one, we looked at the Dimensions COVID-19 data set recently, 17,000 publications, all on COVID-19, and we can have a look at trends. Nearly all the trends were going up, except one which was on Wuhan, and obviously Wuhan is slightly old news, and therefore people are not using Wuhan as an example. So you can see trends and things like that. Another one, this might be more controversial. We took all of the publications from the 15 Scottish universities which have public databases, you know, Pure, etc. And we modeled these, and it's interesting. I've just picked out the brain topic there. And what you can do is you can click on these and you can see what distributions the different universities have. And it's quite interesting. It kind of, I mean, Dundee has a lot of medical stuff and et cetera, et cetera. It kind of reinforces your own views, but there's a few surprises in there. Here's one we've done very, very recently. It's the one I was talking about earlier, where it's the UKRI COVID-19 grants. And so we've done this for them, both in terms of the Innovate grants and in terms of the research grants. And here's the topic map here for the research grants. And they would have had great, I don't think they would have, well, the only way they could have done it without this tool is to do it manually, actually to go through all the COVID-19 grants, decide on a new taxonomy, and then classify all of these things. Okay, so just to summarize here, topics document allocation maps always generated completely automatically from the author's words, all right? So we don't put any words in. The only thing we do is we choose the number of topics. Everything else comes from the documents from the authors. So it's very politically agnostic. It provides a very rapid overview because you can look at it and the way it's organized, the way the maps are organized is organized into groups. It gives you a very, very rapid overview. It's very valuable, we found, for community building, network building, and horizon scanning, etc., etc. We've done it for comparing, you know, how does UK ESRC compare against its equivalent in the US? So trying to spot holes in terms of strategies. But it's this use of fingerprinting documents and sections, and maybe, so what I'm looking at, as well as providing a network with these horizon scanning capabilities, etc., etc., to also be very interested to see how useful it is for analyzing clinicians' notes and turning those into vectors and augmenting the image processing that you would do on your existing data. So it gives you a tool for analyzing unstructured text, and that might be very valuable information which is in there. Okay. So thanks to a whole bunch of people, and I hope I've hit the time, Christian. I think you're still moving.
Yes, here I am. Thank you. Thanks very much, Mike. And indeed, we are right on time. And that brings us to the final presenter in this session, Dr. Graciela Muniz-Terrera. Excellent.
Can you see my screen? Good. Is that okay? Can you okay?
Looks good to me.
Okay, right. So I'm Graciela Muniz. I'm a lecturer, senior lecturer in biostats and epidemiology, and to be honest, after listening to the previous speakers, so their presentations were super fun. So I hope I don't disappoint the audience with my presentation that goes back to the data challenges and opportunities in dementia, particularly in dementia prevention. So you've seen these numbers before. In summary, Bettina and Jerry presented that, and they explained how dementia is a massive public health problem and that there's no cure. That's a big summary of the situation. It's a very bleak situation we are in, and the focal point at the moment, given that the lack of advances in the pharmacological treatments, we are all focusing, or a large group of us, of researchers, are trying to think of dementia prevention as the way forward. And one of the critical questions in terms of dementia prevention is trying to identify those individuals who are at higher risk of developing dementia. Now, if we think of where we are in terms of dementia prevention research, one of the biggest challenges that we are facing is the data availability. Right? What type of data we can use and we regularly use to develop our analysis, to develop our models. As in every other area, we need large enough, good quality data sets. And say, like, many people say, okay, fine, you need large studies to do to do research in genetics, but that's not the only context where you need large studies, right? We cannot keep analyzing data sets and making big statements from data sets that are, from 50 individuals. So we need access to good quality, large enough data. But this, and that is partly where the challenge starts. And because partly the challenge is that collecting new data is not quick, it's not easy, and definitely not cheap. So just to give you some examples of what I'm talking about. From our side, from the side of those who are collecting data, you want to study, for example, in the last study I was involved, the cost per person, per visit, was about 3,500 pounds per person, per visit. And that is without, so that is just the data collection, paying for the time of the psychologists doing the cognitive testing, the nurses extracting the biomarkers, but it's not including the fancy analysis that happens in the labs when the samples are shifted and are shipped and then analyzed. So, and that is, as I was saying, per person, per visit. But then we need the participants, best case scenario, what we do is like do a longitudinal study, so we repeat this assessment over time. So that multiplies the numbers significantly. And as Jerry and Bettina were saying, when they show these beautiful graphs, we need longitudinal data over a number of years So we need to give these studies the time to mature so the individuals evolve through the different stages. So as you can see here, that long-term data collection is seriously expensive. Then we have the other side of the coin, if you want, that we need the participants' perspective. And that's something that is, if you want, in the old times, that was not so, participants were not that involved or as involved as they are now. Right? But we have understood, we have taken on board their perspectives, and we keep doing that. And it's a massive effort towards the continuous involvement of participants into research. And for example, I was involved in one of the in one European study, the EPAD study that I mentioned here, where that was a fantastic example of participant involvement, where the participants in the study attended the General Assemblies, the scientific meetings, and they gave us feedback about the science that we were doing, but also about all the things that were working for them and they were not working for them. And then I have to say that we have made great progress, but at the same time, there are some individuals who still have concerns about participation, and they are still reluctant to be involved. And unfortunately, that is also the case in some, for example, in some ethnic minorities, where the participation of some specific groups is not so common as in other ethnic groups or cultural, cultural differences exist. So they are not so willing to participate. And therefore, when we want to do research and generalize our findings, that can be an issue. So they say, okay, fine, right? We have all these problems when we are trying to access data, to create or collect new data sets. But then you say, okay, fine, right? Suppose I have the data. What's going on in terms of data analysis? And particularly dementia risk analytical models. It's a fantastic area where we think, well, we should be further, more advanced in terms of dementia risk modeling, but unfortunately we are not. And that is something I recently reviewed. I published a review with my team on statistical methods for dementia risk prediction. And we produced recommendations for future work. And actually, it was shocking. It was absolutely shocking, because the analysis employed are basically three different ways, three different analytical methods: logistic regression, Cox modeling, or some sort of more like if you want the data science or approach. Logistic regression, clinicians tend to use logistic regression analysis. There's a very famous index called the qEEG index that's developed from a logistic regression. Everything tends to be cross-sectional. There's no death is not taken into account. So it has flaws from whatever perspective you look at. Cox modeling the same, right? Even though there are, it's possible to do some sort of computing risk analysis, most of the time it's all death is ignored. The proportional hazard assumption is very unlikely to be fulfilled. And then again, most of the times the predictors are just static. There's no, these methods, these modeling approaches are always, they don't take into account the evolution of the individuals, even though techniques exist, they are not commonly used. And data-driven approaches, again, there are a few issues with the methodology used that I'm going to touch in the next screen. So then we have, I also wanted to direct your attention to this other paper where we said, okay, fine, there's a lot out in terms of the mental risk modeling, in terms of specific publications, right? So people grab a data set, they go for their favorite methodology, and they produce a paper out of that. But then there's a lot of noise and very little integration into what we really know. So I think that's an aspect where, as a community, we definitely have to work together. So this is partly what I was talking about. I want to touch this one in particular. Right? The data challenges. Within the community, there have been a lot of a move towards data challenges, and they are fantastic. But when the last one that I am aware of in the mental research is the tadpole data challenge. And there's a publication from early this year where they summarize the findings. And what's really interesting is that there was several international teams of people involved in research and data scientists submitted their approaches. But somewhere there's a paragraph in the paper highlighting how even all these super clever people use their best technique. The limitations of those approaches are exactly the same limitations of the simple analysis that other people have done in terms of, no external validity has been done, no internal validity, the data, the findings are not transportable, have not been double checked. So there's as a community, we are, if you want, we are stuck. Right? We are looking at things from different perspectives. All of them are limited. And the most importantly is that we look at things in unity from a single point of view. I say it's like we don't integrate data sets, we just look into the mental risk, looking into one particular variable rather than, so like in a model including, I want to analyze, understand the mental risk and the role of gate speed in the mental risk, and I go with my model, and someone else is interested in some other variable and they do their modeling without comparing with mine. So there's a lot of noise in the mental risk prediction, but no integration, no overall perspective of what's going on. So I just wanted to highlight the fact that we need to combine more modern techniques with, so we have to modernize our practice in terms of research. We keep looking things unidimensionally, and using still very limited modeling approaches. And there are some initiatives like the Brain Health Scotland initiative that is aiming to get data together, and some of the initiatives that we are as a team, we are involved in. And I think that is the way forward, because otherwise we keep doing what is fun, right, for us, what we are interested in, but we don't seem to be making the advances that are necessary so that our research benefits the us, at least in terms of the mental risk prediction. So there's a clear need to shake up the system, and that's where, as a group, we are all involved in trying to make some advances. So that's it from me.
All right, thank you very much, Graciela. So let's see. Well, I'll just keep talking. I think that leads us really nicely into an opportunity to have an open discussion, since you've presented us with a challenge for the communities to collectively rise to, I would say. I am going to begin by quickly presenting the slides that I thought I was presenting at the beginning of our session. Apologies again. Just so that those of you who are watching can see the links that I referred to. And I will also say that when we go into the open discussion, you should have the option, I believe, to raise your hand if you wish to be called upon to speak with our presenters today. You also have the option of typing a question into the little chat window within our session, which I will then moderate along to the speakers. So you can have some questions brewing just now if you want to give it some thought, and I'm going to try again to share my slides. Can people see my slides now? That's much more like it. So here I was just talking to myself at the beginning of the session and without anything for you all to look at, but this is what I thought you were seeing. This is the map of the Scottish Research and Innovation ecosystem, and the link that I referred to is at the bottom of the page. This has been put together collectively by the research pools, and it just launched at the beginning of last month. So I was introducing myself as the lead scientist for Sinapse, the medical imaging research pool. And since then, we have heard presentations representing life science research, preclinical and translational work from Professor Platt, and data science work from the Sicsa research pool presented by Professor Chandler. And then also in the aligned larger community with other organizations, we have the Scottish Dementia Research Consortium, who Graciela is representing with her talk about modeling and actually having some clinical impact. And this next set of circles is just calling attention to the wider community of innovation centers where there is, I would say, good opportunity for continuing to build connections. And this was how you were meant to have your introduction to the speakers. So we've heard from Jerry, we've heard from Bettina, we've heard from Mike, we've heard from Graciela. And so now you know their names as well as their faces for discussion to follow. And we do indeed want this to be an active, open discussion. So here are some provocative questions, let's say, to get people talking. And this is the link that I meant to show you for our new mailing list, which has launched. So all of the interested members of the audience today are very welcome to register to join the mailing list, and we can continue these discussions beyond this afternoon. So we have, let's see, just about half an hour for discussion. I will, let me see what I want to do. Still see my slides? I was going to start by reading a question that popped up in the chat, which is to all of the speakers. It is from Emily Ann Nault. And she said, Bettina Platt mentioned that effective treatments remain elusive. I was wondering if she or the other speakers had any thoughts about cognitive rehabilitation practices that have been shown to be effective at postponing cognitive decline, although it is often lacking in the literature and in practice. Any thoughts about cognitive remediation, cognitive rehabilitation practices?
I mean, I can briefly comment, even though it's probably more a clinical question. The problem is if the undergoing neurodegeneration is not stopped, you may see some improvements with these kind of therapies, but you are not going to achieve a full recovery or the likes. So unless we can stop the disease process that is underlying, or it's not going to be a cure as such. Jerry, maybe you want to comment.
Yeah, I mean, basically a similar kind of thing to as Bettina's saying. I mean, it is always a bit of a bone of contention because clearly the pharma want to fund pharmacological interventions, whereas a lot of NHS now is moving towards kind of prolonging health and having, you know, things like non-pharmacological interventions. What we're not aware of at the moment is how much and for how long you would need to continue that, because it's not, you know, it's not cost free. That, you know, you need sometimes specialists to deliver it, and sort of individual plans to help that move on. And we're also not clear how it would play into the So say if there's an underlying plasticity component to it, how different mechanisms of neurodegeneration may be suitable or not suitable for that. But it's a perfectly valid thing to consider, and it has uses in other processes as well. But as Bettina says, ultimately what we're talking about here is brain failure, and what we would like to do is, rather than managing the decline, is to try and intervene to actually stop the process that's bringing about that brain failure.
But what is, however, quite clear is that, for example, dementia patients who are in care homes where they have a lot of stimulation and a lot of environmental enrichment have a much slower disease progression in general, as much as you can generalize. So it is certainly the case that just giving people drugs to basically calm them down and sometimes deal with their psychiatric symptoms and aggressions and so on has a detrimental effect. And if you can engage dementia patients in activities, it has definitely a positive effect. So I think the data on that are very clear.
Yeah, but also I just wanted to make a point in terms of modeling approaches. Many times when you look at the evidence is a comparison of between person individuals, between person data. So it's like someone who is taking up an intervention compared to someone who hasn't been involved in the intervention. But it is important to, or it would be important to consider the within person information. So how that, at the individual level, that person maintains their function once the intervention is applied, and to do that in an optimal way, you need repeat measures, and that's hard to collect, and it also needs somehow you need to take into account from what level of impairment that person has started the intervention, right? Because it's possible that intervention works for individuals at a certain level, but not for others. Perhaps if the person has already declined, go along the slope too far, then the intervention will not be so effective. So, but to do that is that's exactly the type of question that is important to understand with good long-term data.
Okay, thank you. I haven't seen any other questions come in through the chat channel, the Q&A functionality there. Can I ask our tech support team if there is a way that the delegates are aware of to raise their hand to participate? Because I'm not quite sure how it works from their end.
Hi there, how are you doing?
Hi.
Yes, on the participant part of the page, there's a raise hand section. So those who are participating on the website, just underneath your face, there'll be a button which is basically for raising a hand.
Okay, and am I meant to see it? Because all I see is a Zoom window, I'm afraid.
For you, you won't see that.
Okay, but do you have an eye on it?
Yes.
Perfect. All right, audience, don't be shy. We have neuroimaging, neurobiology, data science, and modeling experts awaiting your questions and your challenges.
Christian, while we were waiting, I was struggling to put something into the chat window. I only saw the private chat window, but I've got a slightly provocative question. Is how do we stop data scientists from overusing the data? It was provoked by Graciella's comment.
Well, I actually forgot to make one of the comments that I wanted to make, right? So when I did my review on dementia risk prediction models, one of the striking findings was that I can't remember the exact numbers, but suppose there are 230 something papers, right? The dementia risk prediction models. It was something like 75% of them had been developed using exactly the same data set. Right? It's a super famous data set called ADNI, right? And I suspect that the reason for that is that, well, it's a really good data set. I'm not taking away that, but it's open access. And the fact that it's open access, I think it's a key factor for the overuse of the data set. But what is the impact of that overuse? I don't know, and I don't think anyone has looked at. So, but I'm certain we are inflating our results, our inferences, by overusing that data set. So yes, it's a key problem. And partly that's why we need, as a community, and partly what Bettina was saying about this idea of facilitating data access with good documentation of those data sets and a good understanding of what are the features, the characteristics of the data that we have, and even the timing of the collections, the instruments that we have used to collect those data sets. For me, that's one of the key aspects that we are lacking as a community to advance the work that we want to do.
And just to add to that is,
Jerry,
oh, sorry.
I was going to say Bettina had virtually electronically raised her hand, and so I was going to call upon her, and then you can speak up once she has joined. Thanks. Okay, Bettina.
I'm quite proud of myself because I found the raise hand option. Because it's different depending on which platform you're using. So every time I go like, okay, it's always in a different corner. So I found it and I wanted to use it, but I also wanted to say something. I'm slightly surprised by the term overuse, because that was the purpose of ADNI, to be used. And to me, it was clear right from the start that it would generate quite a heavy bias towards that particular data set. And I think that is actually a massive problem with what we're discussing here in general. It's the data bias and the lack of negative data publications in general, also for what Mike is doing, because he is then developing groups or whatever the specialist term might be, but a lot of people don't publish their negative data. And if you don't have a clear signal in an imaging study, or if there's no clear outcome, it would just disappear in some drawer. So we have a major problem with bias in data sets, and also how do you select your participants, which participants volunteer to take part in the study. There's a massive bias here. And I think this is one of the biggest problems we are facing when we're talking about big data and developing new platforms. Maybe somebody wants to comment on that.
I have it.
Go ahead. Go ahead.
Sorry,
and then we'll hand back to Jerry.
I didn't do it virtually, just raise it. I call it a network of longitudinal studies of aging, mostly they were international, but a good bunch of them were from the US. And for example, one of the biggest challenges that we had when we were trying to understand the data sets and to do some analysis integrating the different studies, we realized that, for instance, everyone had gone for their favorite cognitive tests, and then each of those cognitive tests had a different psychometric properties. They had different questions. They were not transportable in terms of the different cultures. They had incredible biases in terms of the participants they recruit. So, we can think, oh, it's easy. We can pull the data together. We can integrate data. But when we go down to the actual data sets, it's really hard. And I think it's a part as a community, but also from the perspective of funders, we have to really engage with funders to support the methodological developments that are necessary to make robust inferences. Because from the funding perspective, methods developments are not that hot, if you want. So I think that's another aspect that we have to work together to change the mindset.
That's great. It sounds like a call to action. Jerry.
Yeah, just to kind of highlight as well that we're talking thus far about data sets such as ADNI that have been collected in quite a controlled way, albeit in different centres and in some cases different countries, but they've at least tried to harmonise things to a certain point. And ultimately what we want to do is to deploy these things into a live fire healthcare setting, at which point the noise and everything, you know, the vagaries of, you know, having different people acquiring the images, different platforms, someone buys a new scanner, that just, so any system or any model that you've got that may be noise sensitive, the noise characteristics of actual clinical data will probably be detrimental. So building in sort of resilience and sort of measures of uncertainty are quite useful, so that you don't have a model that works beautifully on a well-curated data set, and as soon as you try and externally validate it, it fails. I don't want to sound like a counsel of despair there. It is a tractable problem, but you just need to be aware of that as you design and choose methods that noise is an important feature of the exit strategy.
I'm going to hand back to Bettina, and then I see that Yashar has his hand up, and we'll turn to him next.
Yeah, just in the same context, I mean, one of the biggest problems really is also to secure funding for sharing and networking platforms. Because obviously for me as a researcher, I would be happy to contribute data, but somebody has to offer and technically support a platform that makes sharing and networking possible. If you get a 3 or 5 year grant, you can set it up, you can develop it, but what happens after? It will die a slow death because nobody's paying for the maintenance. So this is something I've seen with multiple networks I was involved in, and it's a real shame because we invest into expertise and then we let it die. So I think that's a problem.
Okay, Yashar?
Hi, I just want to quickly introduce myself. Yeah, my name is Yashar Moshfeghi. I'm a Chancellor Fellow at Strathclyde University. So my research is actually sitting nicely in the line of neuroscience and data science. I've been working with fMRI, EEG, TDCS, and so on, and trying to use these techniques for understanding people's interaction with search systems and trying to understand the key concepts that are important in the data science community. One of the things that I think, in addition to the data and sharing the data, I think one of the other issues is a sort of pipeline of processing and analyzing the data, especially neuroscience data. And that's sort of the variations in how people and different groups are analyzing the data, different techniques they're using to clean the data and process, for example, EEG signals and so on. That causes a lot of variation in the output as well. So as a network, one of the interesting things that we can do is to kind of standardize this and provide a unique platform. Obviously, there are some, parts that has to be done visually, but there are lots of parts of it can be done automatically, and we can actually provide such a thing. At Strathclyde, in my group, we're actually initiating this. We are putting together a platform for analyzing EEG data with William Macken, who is also in the chat now, right now, and we are hoping that soon we can actually share that with the community. This also goes the other way, right? There's a lot of packages and there's a lot of sources for data science community that can be put in use for neuroscience community if they want to do predictions and clustering and so on. For example, Mike has shown his work on visualization, but there's other works that can be done in terms of using deep learning models or predictions and so on. So I think that's another issue as a community, especially when we're coming together under this umbrella, we can actually look more into detail into that.
I think that's some really good points. I think along with the sort of standardizing the pipeline, you have to have a protocol, a test protocol by which people can submit their results or submit their algorithms or something like that. Otherwise, what happens is you get this overuse of data. But unfortunately, that's very, very expensive, and there aren't very many incentives for individuals like myself or Yasha to do that, because if we were to set something like that up, we can't compete in our own competition, which is a bit of a shame. But one of the reasons why the data gets overused is because there's no protocol, and there's very much a culture of, I think, allowing PhD students to do multifold cross-validation. And what this does is that the PhD students do it again and again and again and again, and they don't realize that they're overfitting, they're training through themselves. And that boosts the results. And then certainly in the computer vision community, you have to have a good result in order to be published. And so if you don't have that advantage of overtraining, you're not going to hit the top levels. So you do notice within different communities that they treat these protocols differently. So I think, for instance, natural language processing, some sections of that are very, very aware of this problem and put in place and have mechanisms for this. But it's very, very expensive, and it needs government support or research council support, because the standard research grants that you get don't allow you to do this. And I think Bettina's point about, even if you get a research grant to do it for a while, or a network grant to do it for a while, the network grant stops. And that's a real big problem. And actually it takes a lot more resource than is normally in a network grant. So this is maybe something we need to lobby the funders about, both the Scottish funders and also the UK funders, because this is a problem that goes right across, I think, funded science.
Thank you for that point, Mike. It actually ties in nicely with a question that has come up in the comment area related to the sorts of things that funders ask for. The question is, on the data sharing issue, what is the role of data management plans? We are supposed to include them in all proposals. Does anyone have experience of them being effective?
Sorry, I shouldn't laugh.
Thoughts?
They're very expensive to implement properly. That's the problem.
So on one of our European grants, we have extra funding for the data management. And as Mike was just saying, that's really expensive. So there's companies who offer that. And obviously, it takes a chunk of the funding. And when the grant's finished again, what's going to happen? I don't know.
I think there's also a significant problem in deciding what data to archive, because obviously you're not going to archive every single byte that you've changed in the lifetime of the project. But the problem becomes, how do you test whether or not your results are reproducible from the managed data set that you have archived? And the emphasis on the data management plan is not verification like that. So often what we do is we just slam the data in and we forget about it, and we've done the job, but we never actually validate that we can reproduce our results on that particular version of the data set which we've slammed in.
It's something interesting has come out. So I've been in discussions with so my other research interest clinically and academically is brain tumours, and CRUK, Cancer Research UK, have sort of come to understand some of these problems, because there's a lot of data intensive techniques getting used in cancer follow-up and diagnosis, and they've joined up with a company to effectively provide a one-stop shop. So rather than you going out and costing various things and trying to put it into your grant as a line item, CRUK effectively give you a very low friction option to, tick the box to use a sort of data, they call it a trusted research environment. So it's TRE, so you can do computational things, cloud-based, and do some sort of intermediate and longer term storage for, serving it up down the line. Now, it is very much just to get you into the system. So you probably will have to look for additional funding to maintain it for a very long time, but at least it's showing that the funders are getting, behind the concept that your research will generate data and that we need to facilitate where people can handle that and deal with it.
To be honest, I think it needs to be a government level initiative, and it needs to be a government level initiative to stimulate the funders, because the funders have their hands tied as to what they can actually fund. And if you take the research councils, then it's very much science that they're funding, not in terms of immediate research. It's not long-term establishment of resources which will facilitate research, which is nuts. Anyway.
And can I add something? I would like to throw a hot potato on the table, but but you know, I like doing those things. But the other thing is that I think we should not forget is how research is rewarded in terms of career progression.
Yeah.
So unfortunately, when people collect data by sitting on their data, they get authorship. Therefore, there's more, you know, or they copy eyes on grants, and then it's a boost to the CVs. And even if it's publicly funded data, that data hardly ever becomes open access. So, and that's, you know, that is unfortunately is common practice, or much more common than we'd like it to be. So, you know, in terms of lobbying, I think we have a long way to go, and lobbying funding institutions from for many different unfortunate situations and practices.
Yes. So Graciela, you're saying we just have to affect the policy of Bayes, FSC, and REF.
just that.
Yeah, that's all it's going to take.
So who's going to run for Member of Parliament then, and then escalate it through the...
On the very topic of lobbying, a final comment has come through, which I'll read, saying that the National Physical Laboratory is looking to establish a data metrology activity. Might that be a place to direct lobbying towards? And there's a link here to a PDF, I suppose, with more information. I'm happy to share that in the main chat window for today's session. It's news to me. Is that something any of you have heard about NPL?
That actually, I think what we might be able to do is get the Scottish innovation centres together, because all of the innovation centres have this problem with data. Yeah, they all have this problem with data, and they're all struggling with it. And if with one voice the Scottish innovation centres could raise this issue, certainly to start off with in Scotland, then we might actually have some lever, we might be able to change things then.
Good point. I see that Yashar has his hand up.
Yeah, I just want to point out about the data and the grants. One of the good things about the data management plan is it kind of enforces researchers to actually look at the existing test collection that are available in open websites and make sure and guarantees that the researcher is not going to duplicate similar data sets, which I think is kind of valuable. So the whole process of putting in place data management for the current researchers in UKRI is positive in my point of view.
I agree, but it's still quite I think Graciela's point was that the way that, for instance, REF is set up, it rewards publications. That's direct money to universities in their QR block grants. And that then shapes career promotion. And therefore, it's much, much more effective for an individual researcher to concentrate on publications rather than to, for instance, concentrate on impact, or alternatively on making large amounts of data usable by the rest of the community. And I think it's endemic. And this is a discussion which has been going on for, I don't know, maybe 10 or 15 years or 20 years or whatever. I think REF has changed in terms of its attitude to impact, but I think it needs to change in terms of maintenance of resource such as data. And the associated protocols, and refreshing the data so that you have new blind data periodically, say once a year or something other, to do your tests and things like that.
Great. Well, this has been a really interesting discussion. It's ended up broadening out to data science issues for the entire field, not only those of us who are interested in brain health data. I have been asked to wrap things up, as in 5 minutes there are going to be PhD lightning talks starting at 3 o'clock on the dot. Any final comments? Any final burning points to be expressed?
Kristen, is there somewhere for this session that you can put a bunch of URLs and papers for people who are interested?
That is a question probably for the tech folks. What I have identified stumbling through just this afternoon is the chat area. Will has his hand up. Is that regarding?
It was just one more comment about
thanks,
that. And it was just if people have small data sets, then surely they could look towards things like OpenNeuro or Xanadu, for example. And I don't know, I think they look like they'd be useful places to share data. And OpenNeuro, I think, it encourages a standardized format, for example, to share neuroimaging data. So I think they are interesting resources. I really like the idea of a kind of Scottish-wide initiative across all the different research pools.
Yeah, thanks, Will. You were breaking up a little bit there, but you were talking about smaller neuro data sets and how they can be shared with the standard format.
Yeah, maybe the mic's not working overly well.
It might just be the connection.
Is that any better?
Keep talking.
Sorry, is that any better now?
Oh, you've gone quite quiet.
I think I could hear some of what I was saying. So the OpenNeuro thing, and initiatives like DataLad and BIDS, and trying to get metadata into a nice format. They're excellent, and they're designed specifically for these kind of purposes. And the two things I would say is, one is that we perhaps want to expand it so that it can take additional data types, which is obviously worth engaging with the people that set them up. And the other aspect can sometimes be around a lot of NHS trusts in particular, even if they're attached to R&D sort of sections in universities, they basically take the kind of the safest option, which is just don't let any of the data go anywhere apart from specifically sanctioned and whitelisted data havens. And that can be a bit of a problem with, it's easy enough under GDPR to anonymize things and put them onto things like, OpenNeuro and use DataLad to curate that. But if you want to maintain some of the linkages to allow very rich data sets to be brought together, then we sometimes hit into some barriers, which is why in the past we've ended up engaging with commercial entities, because once they've provided a service for an NHS trust, for example, they then become whitelisted and it becomes a path of least resistance to use them again. But I do like the concept of, even if you do deal with something in one way, we should be able to, it's like FAIR principles, isn't it? We should be able to put it onto another thing for longer term storage and sharing out.
Yes. So 1 minute warning. Let's wrap this up. I'd like to thank our speakers. Thank you, audience members, for engaging with us today. And the things that have been identified as challenges needing to be addressed are exactly what we'd like to collectively move forwards to do. I will put all the links that have been shared that aren't yet in the chat section of the conference page into the chat section of the conference page. And on the Sinapse website, I'll do my best to archive the list of resources for longer term access as well. And please do sign up for the JISC mail list to continue today's discussion. Thanks very much, all.
Bye.